All right, everybody. Well, good afternoon, and thank you for joining us for seminar this afternoon. Just to keep us all on the same page, we have 3 weeks of seminar coming up. So Jan, who I'll introduce in just a second here today. Next week is Dave McNear, and then the week after is Trey Canaster from the Center for Education, Teaching, and Learning Center, something along those lines. First 2 weeks of March will be a break, which we're hoping to be interviewing for our extension soil science faculty at that point in time. And then I think we start back up mid-March, and then we'll have them weekly until we get to the end of the semester. So we've got a little bit of hit and miss here at the beginning, but then we should get pretty consistent as we move on. It's my pleasure today to introduce Professor Jan Frouz. So Frouz, say it again, Frozen help. Thank you for that. Professor Jan Frouz, there we go. He studies agronomy at Czech Agriculture University in Prague and biology at Charles University, also in Prague. He holds a PhD in entomology. He had several postdocs in Italy, Germany, and the United States, including a Fulbright fellowship. Recently, he became professor of environmental science and head of the Environmental Center at Charles University in Prague. And at the same time, he became the director of Institute for Soil Biology and Biogeochemistry at the Biology Center. In his research, Jan has been passionate about ecosystem restoration in post-mining sites, the role of soil formation in ecosystem development, and soil fauna microbial interactions, the role of soil biota as an ecosystem engineer, and their role in soil formation and nutrient cycling. He's published over 300 scientific papers in 250 journals, and his H-index is a 44. So join me in welcoming Jan.
Looks like it's on. So hopefully even people online hear me. And yeah. Okay, so thank you for coming. And I will be speaking today about basically interactions between plants, soil biota, and soil, and how this interaction can actually trigger the ecosystem development. And you know, the picture here behind, actually, it's a drawing of successional changes. And this will be actually the scene for most of my, most of my examples today. So
just click on the mouse and it should, there you go.
Yeah. So basically, this is the outline what I will be speaking about. You know, how the plants can interact with the soil biota, how the soil biota can feed back to plants, how they can change the soil, and how this can feed back to plants, and all that is certainly affected by these other soil forming factors, which you all know, like climate, topography, geology. And basically, if we want to tear apart these interactions, we might, if we want to kind of tear apart these arrows, we can see this interaction between soil biota and plants as a one possible pathway. So you can have mycorrhiza, pathogens, rhizosphere microbes, and so on. Then you might have plants directly affecting the soil. You know, the plants are building the root channels. The root channels, where they are abundant, they can be reused by plants. They can be basis for formation of soil aggregates, and so on and so forth. And certainly some soil biota, like, earthworms, but many other creatures, they actually alter the soil. I will be actually speaking quite a lot about those examples because, as was already mentioned, my PhD was in entomology, and I actually start to work with these invertebrates. And this can feed back to plants, but it can feed back also to the soil fauna. And all this is basically happening over time. And what I want to stress here is that there is, pedologists often study soil and they kind of understand the soil is developing, but they assume that the times for that is rather long, rather long. And then, plant scientists, if they consider soil, if they don't grow their plants in some, hydroponic systems, they usually assume soil as, given scene, and they seldom assume that the soil is changing as an effect of plants. And I just want to start by pointing on this picture. These are two soils. Both of them have about 40 years of soil development behind them. They start from overburden, which was dumped, in mining sites. So at the beginning, there was virtually nothing, and there was just a tree was planted. There was no amendment, no topsoil, anything. And you see this A-horizon development in the pine and alder trees. The sites are actually physically located about 50 meters apart. So they have really the same substrate, the same climate, everything is the same. And after 40 years, by naked eye, you can see tremendous differences in the soil profile. And I want to highlight here the time scale, because basically based on and I will get to that several times, but based on my observations, there is really remarkable change in soil quality affected by, plants or by erosion or by other processes which happen in decade time quite quickly. And we often do not study that because, our grants likes for 3 years. And when we have kernel sequence, we often look on much larger time steps. So actually what triggered these differences between these two profiles? So basically, obviously, this is the plants. Alder is a nitrogen fixer. It produces, low CN ratio litter, which is highly supplied by nitrogen. So basically, and the pine produce, difficult to decompose litter. So basically, what we see here that if we supply easy decomposable litter to the soil grows fast. If we supply hardly decomposable litter, the soil grows slowly. I mean, A-horizon at least, grows slowly. And this also is reflected in carbon. So this soil stores about 4 times more carbon in mineral soil than the pine, which on the first glance looks a little bit counterintuitive, yeah? You supply litter which decomposes very easily and a lot of carbon stores in soil, and then you supply litter which decomposes very slowly and only little carbon stores in soil. That's because most of the carbon it actually stores in microbial necromass. And if we supply easy decomposable litter, the microbes eat it very efficiently, convert a lot of to microbial necromass, and this gets stored in soil. However, this gets triggered also by earthworms, which help in this process. Basically, when we have these sites, we actually have more than these two. We have 7 different tree species, and then we study all kinds of parameters. So first, we actually look at was litter production, and we measure litter fall, and we measure understory litter and root production and whatsoever, and we combine it by different way. And to cut long story short, there's no relation to carbon. But we actually found very strong correlation between earthworm density and carbon storage. And we also found very strong correlation between proportion of earthworm cast volume from total soil volume and carbon storage. So basically, what these earthworms actually do, this is aggregate formed by some other processes, roots, shrinking and swelling in the same site, but without earthworms present, sites which are not colonized by earthworms. And this is earthworm cast. And you see a lot of these tiny dark patches here, which are basically a fragments of particulate organic matter, tiny fragments of the litter, we can say, which are embedded in the clay matrix. And around these tiny fragments, the DOC get leached, and microbes on the clay surfaces can flourish and grow, and then they can form this microbial necromass, which actually form basis for this storage of organic matter. And this is actually nice example of interaction which needs some time. Yeah. So when one earthworms came and produced one cast, nothing would be really changed. But if they will continue to do that for decades, you see over 40 decades, you see this 15 centimeter of a horizon. Yeah. We have a textbook when I was a student, and then say 1 millimeter of soil take 1,000 years to form. Yeah. So most of these soils are studied. They never read the textbook, very, very poor student. So, so this is example of interaction that needs some time, but there is another example of cell fauna. This is elaterate beetle. And elaterate beetle are important herbivores on roots. And there was, again, numerous examples in literature that root herbivory can absolutely change course of succession. And this is one of our examples. So basically, we have these pots, and then we grow 2 species of grass, Festuca rubra and Calamagrostis epigeos. And if there was spot which had these herbivores, the Calamagrostis was going to win. And if we put these herbivores, a Festuca is going to win. That's because the Calamagrostis have more juicy, stolons, which are preferred by these herbivores. And we can, both these graphs are actually common in early successional stages. So we can actually test that in field. We use some insecticide and we eliminate these herbivores. And we see when we eliminate them, we actually support Calamagrostis. And when we leave them, Calamagrostis was more vigorous. So basically, after showing these examples, we can kind of summarize that the plant can interact with the soil either by roots, and there is, as I said, a lot of direct interaction in the root, change the soil, produce root-associated biota, and by litter, which again, support biota. And all these interactions have kind of 2 sides of one coin. They often cause some immediate effect, and also they cause some effect which accumulate over time and produce a legacy. And if I just try to make some kind of overview, so this is like this diagram shows these litter-driven processes, and this shows these root-driven processes. And these are these different plant ways, as I already indicated. So this is plant to soil biota and back. This is plant to soil and back. And this is plant to soil biota and soil and then back. Yeah. And these actually show a time scale. So here is a time when the plant is actually growing. And this is actually, showing where is the strongest effect. So this plant, which is grow here, was affected by, plant which was growing season before, and it affects another plants which will be growing season after. And this would be an allopathic effect of litter, for example. Yeah. But this litter effect on the soil formation, that will actually, the plant which is growing there today would be affected by process which was started by plants that grow them, decades before. And this plant which grow here today would affect plants, decades after. Yeah. So basically, certainly these processes, happen kind of continually, but we can most see them manifested after certain time. Yeah. Because these legacy processes, they often accumulate for small changes. So this one generation often doesn't make change, which kind of count. Yeah. And from this diagram, without even reading this, you can see that these roots have actually more of these immediate interactions, which is sort of logical because these roots, are living part of the plants. They are in soil, so they have all opportunities to, in real time, interact with the soil matrix or with the soil biota, which are already in the soil. While the litter, more support this medium-term or long-term interaction because litter is basically dead plant tissue. So it basically takes some time for plants to, come to senescent. And this instantly doesn't affect that very plant, but rather, the next generation of plants. And this is basically kind of the same picture, just converted in another view. So we have this, litter input and root processes. And this is just the one side of this, what I just showed before or after. And it shows some kind of loops we might see in these interactions, ranging from, very immediate effect like root channel formation, which then affect, all root channel use, aggregate formation, which can affect, soil properties, can affect formation of soil horizons. And this again can feed back to plants in various ways. And this can affect also to these immediate interaction between, let's say, symbionts. So this is pathway that people often study when they speak about plant soil feedback. So you have symbionts or pathogens in the actual generation, but they can accumulate in soil and they might affect next generation of plants. But this can be also affected by changes in soil conditions and so on and so forth. And this is basically, again, the same picture, just showing example of some of these processes and kind of showing this, temporal scale of those, but it also trying to show that these processes need a different space to operate, yeah? So these small scale processes often happen on the scale of microbes and individual roots. So they actually need really small space to happen, while, changes of soil profile really need accumulation of these small processes over larger time, but also over larger area to be effective, yeah? So this temporal actually scaling is associated also to spatial scaling. I will just make few notes about, this soil fauna, because I'm not going to say they are more important than microbes, but they are often neglected. I actually spent a great deal of my life to study them. So I want to kind of point on those. And in recent review we found that the soil fauna overall can consume globally about half of annual litter fall. So whatever fall on ground in the form of that litter, half of that can be eaten by soil fauna. It certainly vary from biomes. It's less in desert and more in temperate forest or grasslands. And you can see here, similar picture about different processes the fauna can affect carbon turnover and carbon storage in soil, which started through processes during the good passage and excrement aging, and then all the way to soil development. And this rectangle here approximately depicts lifespan of the soil fauna. So many of these processes on the left part of the figure happen during the lifetime. It's a kind of immediate interaction, but many of them are actually product of accumulation of this fauna product over time. And I would actually, so we can again speak about some immediate effect and about some legacy on this gradient. And in rest of my talk, I would actually try to speak about this temporal effect, about this accumulation of this temporal effect. And this is actually the very simple experiment when we try to study the effect of earthworms on the carbon storage, because in that previous experiment I show you in this pine and alder, we found that the earthworms actually support carbon storage. We want to mimic that in laboratory conditions. So we have 3 bottles. Well, actually, we have more bottles, but with 3 types. And in the first bottle, we put a liter. The second bottle, we also put a liter, but we crush it and mix it to the soil, which was the overburden, mining overburden, which very low carbon amount, low carbon content at the beginning. And then we put liter on the soil surface, and we have 2 earthworms in each bottle, which mix it for us. And every time when the earthworms eat up all liter, we just, in all the bottles, add the same amount of liter again, yeah? So we add here in the surface, here we mix, and here we add in the surface again. And then we have these beakers with sodium hydroxide, and I have students who like to titrate those, and because they like that very much, so we do that for 2 years, and then we, several times replace this liter, and I'm showing just this first addition after 20 weeks, and then the 4th addition, the last one, which was from 80 to 120 weeks, yeah? And basically, if we look on that first addition, we can say that the respiration in the earthworm treatment was the highest. So there was highest loss of carbon from the system, while this treatment, which we mixed, like imitate the tillage, if you want to say, this actually was most conservative, most carbon was stored. And we can stop experiment after 20 weeks and publish it, and everybody would be happy. But because I have a lot of these students who like to titrate so much, and I actually encourage them to do so. So we found after this almost 2 years, completely opposite trend, yeah? So the earthworms actually now was the most conservative, and mechanical mixing caused the highest loss. So, and this is, this would be a phenomena I will be returning to my talk several times, that this actually is changing. The same effect might have different outcome over time. I will stay in the earthworms a little bit. So basically, this is, successional studies of plants, quite classical. So we have like bunch of sites, different ages from 1 year to 41 years, and our fellow plant community people do extensive survey of plants. And they actually found 2 distinct community of plants. One they called like ruderal plants dominated, and second dominated by late succession plants. And we actually measure all kinds of other parameters. So then we use discriminant analysis and backward selection to get smallest basically set of discriminant parameters. And we found that the presence of humus layer, even thick, which was produced by this earthworm feces, is actually what determine this shift. So if I can kind of wrap this in some story, this first generation of plants allow earthworms to colonize the site. Then earthworms colonize the site, change the soil a little bit, and this allows more demanding species of plants to colonize the site. We did a lot of experiments to test that, and another experiment was here. This is like intermediate stages of succession before earthworms came, and we was actually interested to see there is no understory in that shrubby vegetation. So we use these iron frames, and because I have a lot of students who like to push things by hammer, so they hammer those to soil. And what we actually found that We have two types of those, one have these windows so roots can grow back, and one for solid. And when we put this solid, we see in 1 year, there is some understory already pop up. We actually also tested, the light. So half of those was thinned to have a more light, but this has no effect. But this has very strong effect. And when we look in here, we actually see more arbuscular fungi in there. And this leads, to other exploration. And we actually found that the Salix caprea produce high density of ectomycorrhiza fungi, and this ectomycorrhiza fungi tend to outcompete this arbuscular fungi, and if we kind of suppress them by this framing, we get more arbuscular fungi and more plants kind of depending on them. And then we tested if that earthworms can do something with that. So we collect the soil from these sites. We put half of the soil and we inoculate earthworms there in some large bottles, and we kept them for 1 year. So we let them 1 year to work on soil. We added litter, and the other we kept in another similar conditions, but just without earthworms. And then we put this soil to these pots, and we have, two ectomycorrhiza plants, two endomycorrhiza plants, or ecto and endomycorrhiza plants together, and we grow them which and which out earthworms and which out earthworms affected soil. And again, to cut long story short, if we have this combination in soils which wasn't affected by earthworms, the ectomycorrhiza plants and ectomycorrhiza fungi are going to win. And if we have earthworms, the arbuscular mycorrhizae were going to win, which is consistent with some other results that they like more nutrients. The earthworms actually change also food web. In soil food web, we have this fungal pathway and bacterial pathway, and we can actually see that this is a proportion of this fungal and bacterial pathway. So higher numbers here show the fungal pathway is dominating. The lower numbers here show bacterial pathway is dominating. And at the beginning of succession, we have just this alkaline spoil and not so much life, but everything is dominated by bacteria because the conditions. Then we have this shrubby situation, which is fungal dominated, and then we have earthworms coming, and it's again turned to be more bacterial dominated. So basically, if I wrap it again, so you can shift from this high amount of litter, partly decomposed litter, to soil surface, to the situation when the litter has been mixed, and basically the soil food web turned from fungal dominated to bacterial dominated just by presence of earthworms. However, the earthworms actually, the carbon flow through earthworms in the all food web is almost negligible. Yeah. So it's not because of their predation, but it's because of the engineering effect, because of changing of the soil habitat, not really by direct predation on I mean, this might also contribute, but probably wouldn't be the major factor. They also change other stuff, like they change ability of soil to hold water. So this is water holding capacity, water field capacity, and wilting point. And basically, this is which fauna. So these dark columns are basically affected by fauna, namely earthworms. And you can also see consistently they can have more water in all these three. And difference between wilting point and field capacity, this water which is available for plant, is also higher in the worm treatment. I see earlier this week, much better example than this one, but I will show this one. So this is basically the successional sites I was speaking about, and this blue are actually this green are successional sites, this pink are sites when the coal miners plant alder. As I said, alder is nitrogen fixer. And then we studied how good these sites are for establishing of next generation of climax trees. So my students, take this GPS and go around. These are the same size polygon, more or less the same distances from the source of seeds, which is somewhere there. And they found over 300 seedlings in the succession site and one in this outer plantation. This was, by the way, highly statistically significant. And we actually also do the same for oak. And we also plant the seedlings to alder and oak, and we found there is more ectomycorrhizae in this successional sites. So again, we believe that putting too much nitrogen might kind of hamper this ectomycorrhizae. I was speaking about that the activity of the same agent. So we actually do something, we put earthworm to the bottle and they do something. And if we study them for 2 years, they do something else. And I'm going to show a couple more examples of those. So this is actually the experiment with no tillage. So there was one large field and half of the field was divided in tillage and the second of no tillage. And we just go along this intersection, around this border, this barrier, and we take a monolith of soil and we remove soil fauna from this monolith by freezing. And then half of this monoliths we add fauna and half of it we don't add fauna. And then we see basically how that affects nitrogen acquisition by plants and nitrogen leaching. And basically this fauna can have two effects, more effects, but we can say some of these effects, like effect on nutrient availability, can be manifested more or less immediately for plants. The other effect, like formation of the aggregates and formation of soil structure, takes longer and can be more persistent. So basically we have much higher density of earthworms in this no-tillage treatment. Yeah. So we can say that this no-tillage treatment was, among other things, affected by these earthworms for a long time. But this was disturbed regularly. And if we add earthworms in this disturbed treatment, we actually see the earthworms support leaching of nitrogen and reduce nitrogen use by plants. But if we put them in this no-tillage treatment, we actually found complete opposite. So they support, take up nitrogen by plants and reduce the leaching. So basically, we try to detangle a little bit more this legacy effect and long-term effect. So we have several experiments. So we use this kernel sequence. As I already showed, we have the sites before earthworms came and after the earthworms came. So we take soil from these two sites, and then we can again put the worms there or not. And then we can actually measure difference between this no worm treatment in the soil, which was historically affected by worms and was not. And this is this legacy effect. And then we can add worms or not. And this is actually this immediate effect. And not only this legacy effect, but also size of this immediate effect vary depending on previous legacy or not. And also, this is actually the same experiment. Basically, if we put earthworms and we have more than one species, we have early and late succession species, the earthworm have no effect in that soil which have no legacy. But if we have soil which already have some worm legacy, the earthworms actually promote late succession plants against early succession plants. Yeah. So their effect is actually dramatically different, depends on the setting in which we put them. We actually redo this experiment with similar results in a way, because here, you know, the succession, so a lot of things can happen. So we actually choose now the sites which earthworms didn't colonize yet, and we face some of the pots, you know, by mesh. So we take a bulldozer, we you know, dig a mesh, bury mesh, you know, 1 meter below and 1 meter above, and then we inoculate earthworms all around, and we wait for 10 years, and then we take soil. So we have exactly the same site, worm, no worm, and exactly the same result. So we can be pretty sure, but this is not published yet, but we can be pretty sure that this is worm effect. So when we want to detangle the effect of microbes and the soil structure, which are already there, we basically again took this yearly succession soil and, you know, well, I say late, but this is like 40 years old site. And then we basically X-ray sterilize the soil to get sterile material. And then in parallel sample, we take soil from these two sites and we basically make dilution, and we make two dilution, one more and less concentrated, presumably more and less diverse. So this leads to all kinds of combination. And basically, if we look on above ground biomass, the only things which matter was soil age. Yeah, late succession soil always produce larger biomass than early succession soil, no matter how much, what inoculum we put. In roots, it's already not so clear. Then it's also some prevalence of late succession soil, but you know, early succession soil and high diversity inoculum may have in fact higher root biomass. And when we look on the species, actually, we found a tremendous difference between behavior of grasses and herbs. So grasses mostly reflect soil age, regardless of, you know, inoculation. But the herbs actually like high diversity inoculum, which are these blue bars, and early succession plants don't care about source of inoculum. Light succession plants actually like light succession inoculum. But basically, we can see in this experiment, there was very strong effect of these physical changes of soil over time, which was more important than this microbial inoculation. So this is how this show in picture. So this is actually early succession inoculum, low diversity, and you see grass, love it. And you know, the herbs more like this high diversity inoculum, as I said. We redo the same experiment, exactly the same approach, but we used meadows, the one meadows which was tilled and then restored for like species-rich meadow, and the one which was natural species-rich meadow all the time. And here, the effect of substrate is not so much pronounced. Yeah, we saw the soil was important for some species, inoculum was more important for other species. So, but we cannot really say this profound effect of soil changes. Yeah. So, and I can have more examples like that, but I just not want to wrap it in some kind of conclusion. So basically, if when we have, you know, this early succession situation, then this soil changes and this legacy effect will be probably more important than this immediate effect, which would be more important in like stabilized system, which are kind of soil is well developed. I want to move to more long-term changes along the soil formation I already started with. So this is another kernel sequence. This time is not the mining site, but the landslide. Landslides are good because you can have you know, very old landslide. So we have landslide from 4 years to almost 13,000 years old. Not exactly, we dated them by radiocarbon, so there is some kind of uncertainty, but you know, if it's 1 year older or in 13,000 years, doesn't make so much difference. And we basically see that the carbon in mineral soil level out between 50 to 70 years. So basically, it's quite short time from zero to saturation of carbon in mineral soil, yeah? And then it stays more or less the same, remaining 13,000. There is certainly some fluctuation among the data, but this is it. And if we look on that litter layer and fermentation layer, the overlying horizons, you know, this is logarithmic scale. So if we make it linear, it will be basically a line. It will be increasing over time. I just want to point on this red line. This is phosphorus in the system. So you see the phosphorus over a few thousand years start to get depleted. This is actually the study from US, from Illinois, and this shows, you know, changes of carbon after tall grass tillage. And you can also see quite decrease of carbon, which is again quite sharp, and then it's level out and it stays for a long period of time. And, you know, this is the same picture I already showed. You know, this is this 13,000 years on landslide. And basically, we actually collected data about common garden experiment, which was on soil of various ages. And we found that if we look on the carbon on mining sites, the broadleaves tend to support carbon storage over conifer. And if we look on the mature soil, when there was forest before, at least one generation, the conifers are often better, yeah, in terms of carbon storage. So it basically shows that because conifer stores a lot of carbon in these overlying horizons, so once the soil is already saturated by carbon, you know, supplying easy decomposable litter doesn't make much effect. So we can basically see that there is different processes taking part in different parts of this curve. And this, you know, starts from small scale processes all the way to soil profile. I want to show that not only plants, not only fauna, but also physical processes can have a role. So this is a picture from our favorite mining sites, and you see these waves which were created by this mining machinery, how they dump this material. And far from camera, this grassy strip, you know, the coal miners take a bulldozer and make it nice and smooth. And as a consequence, they promote this grass flourishing. And we actually asked a coal mining company to make for us, you know, 3 pairs of the sites, one wavy, one flat. And we studied them for a couple of years. And we can see that in this leveled sites, basically, the grasses start to be dominated. And in the wavy sites, the shrubby vegetation, the woody vegetation become dominant. This is also seen here. So basically, you can see these white columns are for a wavy site, and this dark for leveled sites, and this is for trees or shrubs which are taller than 1 meter, and you see no of them was found after 12 years in that wavy sites. Because these wavy sites get more compacted, yeah, just not because of leveling, but because of processes that happen due to transportation. So to study these processes, I actually use another my favorite site. So I have to introduce you Falcon Catchment Array, because we like to study this ecosystem processes, these large-scale processes. So I thought, you know, it's really not enough to study them in a bottle. So I need some kind of pocket ecosystem. So I hire a company, and they make me these 4 Olympic pools. They excavate, you know, soil 2 meters deep and seal it completely. So they make impermeable, like a pool, basically. And then we put drainage in the bottom, and drainage goes to these wells, you know, in front of those. And then we have this collector, so we can collect surface runoff and subsurface runoff. And then we put here edit hours to get carbon budget, and we put our 400 sensors to measure everything else and collected in these house. But here we focus so you can see how this looks like. And two of these sites we make a flat and we planted by alder, and two of these sites we make wavy, and like original machinery will do, and we leave them for natural succession. And despite all this massive instrumentation, what I'm going to show you actually depends on this erosion pin, which is very simple tool. So it's a piece of wire. We stick to the soil in a way that this red part was completely on the soil surface. So my students can come and measure how much this red part was exposed, and that's the amount of erosion. And we can actually see very high erosion on elevation and some accumulation of these flat areas. But we also see there is some surface increase in these flat place, almost no erosion. This increase probably is because site was compacted, and then by freezing, the surface little bit pop up. But we also see, and I didn't show the data here, that there was very low infiltration in this depression, but there was also very low infiltration in these flat sites. And the bulk density was much higher in these flat sites and depression, because once this clay was liberated, they basically fill all the spaces, and doing that, this flat sites basically seal and get compacted even without additional machinery movement. Yeah, so you can see on this drawing. And here you have this highly porous material on the top, and this transportation effect, all the physical and chemical processes, properties, but it also affects establishment of woody vegetation, which basically tend to establish in this top part on this northern slope, behind the wind, when the snow accumulates and so on. But also due to the fact that the physical properties of soil has been changed due to topography, which is, obvious, but I just want to show this can interact with plants. So basically, I'm going to see that plants, soil biota, this physical environment triggers some immediate changes, and but also some long-term changes. And if these long-term changes accumulate in larger amount, they can come to some tipping points when the system quite abruptly, suddenly start to change its behavior, like when we have a switch, instead of this gradual changes. And that's it. I thank you for your attention. You might read more in this article, and I would be happy to answer any question.
Thank you. So you got any questions from the crowd? Ernie.
You showed in your landslide study, I think, if I remember correctly, that in the mature forests, the conifers were often better at storing carbon.
No. This, I put kind of two stories together. So this conifers broadleaves, that was meta-analysis study. So we basically collected all kinds of common garden experiments, which was established in different conditions. And these common garden experiments, which was established in mining sites, they tend to show that broadleaves are better. And this, which was established in mature soil, So it wasn't related directly to this, landslide.
I was just wondering if you think that will eventually happen in the mine sites. Will the conifers eventually overtake the alder forests in terms of carbon storage?
We actually see in we have this kernel sequence, and we study carbon storage in these alder sites, and we go them like 10 years after, so we can actually have not only like a kernel sequence, but we can have changes over 10 years related to time. And you can see that these young side have high accumulation, and this accumulation rate in mineral soil decreased over time. And 50, 60 years old sites have almost no accumulation for this alder sites. But if we do the same for succession site or for conifers, we'll still see increase, yeah? So the conifers are basically slower at the beginning, but later they catch up, yeah. So that's the answer to the question, yeah?
I have a worm question. You seem to be a worm man. Maybe you'll know the answer. How do worms manage long distance dispersal?
Yeah.
Do they get carried by birds and dropped around?
Yeah. So basically, worms are generally not really excellent migrant, you know. So as you can see in United States, you know, when you have this Wisconsinian glaciation, you know, worms, for some reason, if you cover them by kilometer of ice, you know, you keep them 10,000 years under ice, you know, they don't like it. And they're poor migrant, but how they can do that? So firstly, birds, epigeic species, they have small cocoons and they can be easily attached to feather and they can be transferred to quite significant distances. Then basically soil, like boring, you know, wild boars and so on. Then some of these larger epigeic species or endogeic species, they can actually, you know, in Europe, when we have a lot of worms, we see them in spring days or autumn days when there is a cloudy and rainy all day. We see them travel over the surface and they can do hundreds of meters by one night, yeah, by that, because they do not need to really, you know, go to soil, which is much more demanding. And certainly, you know, humans are very good in that, you know, all that invasion of earthworms to North American forests depends on the strength of American economy, which is, you know, oriented on services. So if you go fishing, you certainly wouldn't go to your garden compost and you wouldn't make your hands dirty. You will go to Walmart or some bait shop, you know, and you can buy, you know, a plastic cup with the earthworms already made for your hook. And when you finish, you know, you just kind of humanitarian action, you know, you release your worm to the forest. And so that's very nice. That's also very nice, I would say, vector of long distance invasion. And then, you know, horses also can carry cocoons. Yeah. So there's a lot of kind of, I would say, transporters, vectors, which can help worms to migrate.
You have a lot of worms at some of your sites. Those, the density on some of your graphs seems really high.
Yeah.
300, 400.
Yeah, but this is really, yeah. But this for the other forests, let's say this is quite normal in Europe.
Right.
And these are large amount of epigeic worms, so they are small. So you can really, if you just, you know, look in the litter, you see dozens of them just by naked eye instantly.
Do you have the really big ones that go down deep and?
We have some big ones, like some Ololopora, but they're not too abundant. They are not in mining sites, definitely. Yeah. Yes.
Are all earthworm species like the same in terms of these processes?
No. There is large diversity of them, and not all of them mix the soil. Yeah. So some species live only in the litter, and it behaves more like, you know, any like millipedes. I mean, ecologically, yeah. So they basically just break the litter to smaller pieces. Some of them burrow in intermediate soil column, like 30 centimeter deep, and they mostly feed on, you know, particulate organic matter, which is already there. They have slow turnover, but they can actually cooperate with these species on the surface, which makes the droppings, and they can incorporate these droppings to soil. And then you have species which go on the surface and, brings material up and down. And in Europe, they can be responsible for formation this deep molly soil, which can be, meter deep, a horizon. Yeah. So this really large array of species which behave very differently in those aspects.
Are they like dependent on like the forest ecosystem? Like would the same ones like that travel through the litter layer be in both the alder and like the pine sites?
No. That was the point. Pine signs have almost no earthworms.
Right.
If there is just this epigeic species who doesn't really mix the soil. So basically, when I show this first graph, what actually makes this litter to be mixed, this 15 centimeters, this due to this mixing earthworms, Lumbricus Lumbricus rubellus in this case mostly, and then some endogeic like Apodectoria caliginosa, which do this basically mixing from surface to deeper soil layer, and they mix like about this 15 centimeters. So basically, if we came to the site 20 years after, the depth doesn't really change. It grows very dramatically, and you would really need some more deeper mixing species to make the profile more deep. We don't have those there. It might be related to the fact that the clay is not weathered so much deeper down, or there is other conditions. Yeah.
It's the same here. On rainy days, there's a bunch of worms that show up on the cement.
Yeah.
And I've always wondered if they do that because that's the best conditions for them to migrate.
Yeah.
Or if they're doing it because their homes are too wet and they're oxygen starved and they're coming to the surface for oxygen.
Very likely the first is right, because it's you see this worm tubes, at least in that worms that go really horizontally deep to the ground. This is really sealed by clay material and by geosphere. You see, for example, ants, they excavate their corridors by taking soil out. Yeah. But the earthworms, they actually press soil on site. So material stays there, it's just more compacted around the earthworm. So it's like a tunnel, and you like spray it by this concrete, sealing. So basically the same do earthworms. So when this channel is active, it's very difficult to flood it. It's like if you have empty glass pipette and you have it stuck in the bottom and you want to pour water in it, you wouldn't succeed. Yeah. But if you have a flood and also profile is flooded by water, then they get flooded as well. Yeah. So if you have big flood, they certainly escaping. But if it's just normal rain, more likely they use that to migrate.
Well, you guys, I have to go to the next Zoom interview for Extension Soil Science, but carry on. Thank you for your talk and the worms.
Okay. Yeah.
Your mining sites, is it all coal mine or what's the overburden?
Yeah. So all sites I show is a coal mine. We mostly study coal mines. That's why I came here, because we have excellent coal mines as well to study.
Yeah.
And no, actually, I know they was laughing, but I'm not going to advocate the coal mining, but I would be really trying to advocate, post-mining sites for fundamental research, because these post-mining sites are really excellent for fundamental research, because you can make a really large-scale experiment. I mean, if I ask, normal grant agency to make me, 6 hectare plots, half of them flat and half of them wavy, it would be difficult. If I ask mining company, they do that. It's a business, it's just a little bit negotiation and you can have it. So the overburden in our sites is alkaline clay. So basically material is like pH is 8. And when it came, it looks like a concrete. but in 2-3 years, it breaks in small pieces. So it's a clay which is impregnated by calcium carbonate and some other carbonates. Yeah. It's actually one of my students made the study when she found all possible, studies around the world about overburden. And most people thought overburden would be sandy and, or clay and acidic. But actually we found that there's quite a lot of overburden which are heavily gravel, like here, or the same amount is heavy clay, like what I show, and then everything is between. And about 30% is really acidic, and 30% is alkaline. This is most like abundant, like pH 8 or below 5. And then everything in between in smaller numbers. So this is quite typical overburden. And it grows really fast, I show these wavy sides here. Yeah, I show this wavy side here. So basically, this is how the site looks like 20 years ago. And this is aerial picture of the same site today. And this is non-reclaimed, yeah? And it's covered by trees in 20 years just by volunteers. And trees are, taller than this room. So the succession really